Hello everyone, so my name is Diane Saunders and I'm a group leader here at the John Innes Centre in Norwich in the UK. And today I wanted to share with you some of the work that my group's been doing in order to respond to the potential re-emergence of wheat stem rust in Western Europe. And so many of you will be aware, wheat stem rust started to re-emerge in Western Europe around 2013, with a number of sporadic outbreaks being recorded, for instance, in Denmark, in Sweden, in Germany, and also here within the UK. And this was a very significant event as it marked the first recording of this pathogen occurring in many of these countries for quite a number of decades. And also since 2013, we've had a number of additional sporadic outbreaks being recorded, and also some that were much more severe, such as that occurring in Sicily. And then even just this year, we saw wheat stem rust being recorded, for instance, in Ireland. And this was the first record of stem rust in Ireland, we're thinking, since around the 1960s. And so it seems like this pathogen is indeed starting to resurge. And so when we start to think about how we can actually respond to this, one of the key questions that comes to mind is where on earth is this inoculum being created? And so if we think about wheat stem rust, it's really a warm temperature disease. And so the pathogen, it creates these asexual inoculum in order to transmit infection. But for the asexual inoculum to start an infection within the UK, they have to be transported in from warmer, milder climates. They can't survive through the cold winter months. And so that means that they tend to arrive much later within the growing season. But we also have the potential for in-country inoculum to be created through the form of these aeciospores, which are the result of the pathogen completing its sexual cycle on its alternate host, these Berberis species. And so these sexual spores can be created much earlier within the growing season, and of course start then infections quite early on, which could potentially lead on into an epidemic. And we know from work that we've done in my lab, going out and looking at these barberry bushes, we're able to find infection quite readily on barberry bushes in many locations across the UK. And also significantly last year, Elizabeth went out to one particular location, which is located just south of our John Innes Centre, and she found that she could identify stem rust both on the neighbouring barberry hedgerow and also within the barley crop. And so she could show that this pathogen is able to complete its entire life cycle, including both hosts, directly within the UK. And so these barberry bushes, it's thought, were brought to the UK around the Middle Ages, but as early as the 18th century, it's shown that farmers were starting to destroy them. And they were starting to rip them out because they noticed that any crops that were right next door to these barberry hedgerows were getting really infected. And by the 19th century, botanists were already recording that they were quite effective, and they were starting to see removal of these barberry bushes from the English landscape. But stem rust hasn't been a problem in the UK for a number of decades, with the last epidemic in England being recorded back in 1955. So of course they've been increasing in natural prevalence as they're a very popular hedgerow species. In addition, we're seeing a number of conservation programs actively planting these bushes. In particular, Berberis vulgaris is being replanted, which is a very susceptible species of Berberis for wheat stem rust. And this is being planted in order to create habitat for an endangered moth species, this Barbary carpet moth. And so all in all, this has created a situation where we have a large number of these Berberis vulgaris plants all across the UK. And so that then draws us to the question of how do we actually manage the risk of these common barberry bushes and the fact that they could be harbouring wheat stem rust inoculum. We already know from our own work that we can find stem rust readily on these common barberry plants. And so that's where Vanessa, a very talented PhD student from my lab, stepped in with the power of mathematical modelling to try to address this question. And she's done a huge amount of work looking at the risk around these common barberry bushes. And today I'm just going to talk about two areas that she's been working on. And so she looked at how much inoculum is being created on these barberry bushes and how that is released from these barberry bushes. So she's looking at the number of aeciospores that is being created, and also how environmental conditions could be influencing these infections. And I can't go through the wealth of data that Vanessa has actually created. We'd be here all day, but I'm just going to touch on some of the work that she's done. And so the first question was, how many aeciospores are really being created and released from these aecia? And so here we have a picture of a single aecium that's being created. So we're going to call this one infection. And so Vanessa then worked with our microscopy department to do a lot of SEM analysis and then a lot of computational analysis of those SEM pictures. And she was able to show that within one single infection, she could see that we were creating around 250,000 spores on average. And so this is a theoretical prediction of how many spores are being created within a single aecium. So then she went to the lab and she did some other experimental analysis to look at, from this theoretical 250,000 spores, how many of those spores are actually being released. And through a lot of analysis, some of which is shown here, she was able to show that of the 250,000 spores that she had predicted, around 3% are actually being actively released. So around 8,500 spores are being released per infection. Vanessa went on and she did a lot of work around modeling the potential mechanism of aeciospore release, including, for instance, what's shown here, which is high-speed videography, where she was able to then monitor the release of these aeciospores from these individual cups. And you can see aeciospores being squirted out of this aecial cup. And she was able to show that this process was a mechanical process. And so these aeciospores were being actively released by the pathogen. And the release was at a huge velocity. So this was a very fast release process. The initial release was occurring over 0.001 of a second. So just a millisecond it took for the spores to be ejected. And so this is really important knowledge, because this tells us that those spores are getting actively released. And so that means they're getting into the free wind area. They're not being pulled by the gravitational force of the leaf. They're actually getting into the free wind area and being then potentially transported on the wind current. Next, Vanessa then went to the field and she started to look at the structure of barberry hedgerows within the UK and how close they were to adjacent crops. And here we have a picture where the redded dot line shows you where a barberry hedgerow is, and next to that we have 2 barley fields. And she was able to show that within the UK we actually have a position where we have our hedgerows and then about 5 meters of distance from those hedgerows before the cereal crop starts. And so then she could employ Gaussian plume modeling with all the information she gained on the number of aeciospores that are created within these aecia and how they're getting released. And she was able to show that around 1% of the spores could actually be released and travel around 25 to 30 meters. And then she looked at that 5 meter distance that was allowing the pathogen then to get to the edge of the crop. And she showed around 8% of spores could travel just that 5 meters that was needed to start an infection in the crop. Next, Claire then stepped in and she started looking at the viability of these aeciospores, because of course all these spores might get released, but how many of them could actually potentially go on to cause an infection? How viable are these aeciospores? And so to test that, she looked at germination rates. When she took fresh aeciospores from the field, around 90% of them could be germinated effectively if she looked at temperatures between 5 and 25 degrees. So most aeciospores that are released are actually viable. Then Vanessa put all of this information together. She knew that from one single infection site, you get around 250,000 spores being created. Just a fraction of these get released, so 8,500, and then just a small portion travel that 5 meters that allows them to get into the crop, and around 614 spores would then be viable. However, this sounds like a very small number of spores, but if we look at a single barberry bush, and we look at one that's got a medium level of infection, we look at the number of leaves that are facing the cereal crop, we have around 400 aecia. So this again is around 250,000 spores. In this case, these spores are viable. If they land on a nice green, tasty leaf tissue, they will then go on to cause an infection. And so it will be no surprise to many of you that if you have barberry in a hedgerow right next to a cereal crop, this could create a very high risk of getting stem rust infection on the edge of your field late within the season, just as we've seen within the UK in practice. However, what was really nice about what Vanessa did within her modeling was trying to employ this Gaussian plume model, which allows us to also look at how many spores travel different distances. And so that way you can kind of come up with a risk across your field, and you can tie this into how far you should plant those bushes away from your crops to really minimize the risk. And so this then helps us go back to people who are working on actively planting these bushes and give them proper advice based on scientific evidence on how far these bushes need to be away from our cereal crops to minimize the risk of spreading infection. And so in summary, this wheat stem rust pathogen, well, it has been identified in the UK and it's becoming an increasing threat in Western Europe. And so we really need to take this seriously. Also, stem rust is frequently found on common barberry in the UK. It can easily spread from barberry to an adjacent cereal crop if the conditions are just right. And modelling aspects of spore dispersal can be very effective in trying to assess risk and guide and inform policy on safe distances for planting of these bushes. And what's really great is Vanessa has also developed all of this work into a web-based model, which we will be releasing in the next few weeks, we hope. And so then you can put in your own location data and you can work out what the risk is at different distances from an infected barberry bush. Okay, so then I'd just like to thank everybody that's been involved in this project. I'd particularly like to thank Vanessa, Elizabeth and Claire, who are our team Stem Rust in the lab, and of course everyone else within my lab who has contributed to this project. And I'd of course like to thank our funders and all of you for your contributions and attention. So thank you very much.